In a handful of cutting-edge laboratories today, researchers are constructing functional, microscopic machines that are actually biologically alive. Historically, we have defined robotics through the lens of engineering, systems built from metal and plastic, operating on the binary of rigidly coded logic. But an entirely different approach to robotics is emerging. These researchers are assembling autonomous systems using nothing but self-organizing living cells. This is the neurobot. It is a biological construct with a primitive nervous system, allowing it to physically explore and react to its surroundings. The most striking part is that these behaviors weren't pre-written in a genetic code. By providing the raw materials and letting the cells organize themselves, scientists have created a machine that effectively makes its own decisions. This process starts with the African clawed frog. Scientists isolate unprogrammed stem cells from the embryos of this specific species, Xenopus laevis. When left in a simple saline solution, these cells don't die. Instead, they spontaneously self-organize into small, spherical structures. These constructs move by repurposing their own biology. They use tiny, hair-like projections called cilia to paddle through the water, propelling the entire structure forward. These first-generation xenobots were limited. Their movement was dictated strictly by their physical anatomy and the laws of physics, lacking any internal control or central decision-making. While biologically active, they functioned like organic clockwork. To move beyond simple physics, the team had to attempt to install a control center. As these cellular spheres were forming, researchers injected them with neural precursor cells, the raw materials needed to build a brain. These cells matured autonomously. They extended branches called axons and dendrites, forming an interlocking neural network without any human guidance or external programming. The cells communicate across this network through bioelectricity. By using specific voltage patterns, the collective can solve problems and synchronize its movements. Proof of this internal control appeared when the bots were exposed to a seizure-inducing drug. Their movement changed instantly, shifting from simple swimming into complex, repeating spirals. Genetic analysis even showed the bots were activating genes usually reserved for vision. Without eyes or a traditional body, the organism was attempting to perceive its environment. By providing the right environment, scientists didn't just build a tool. They sparked a self-governing intelligence that operates independently of its original engineering. This technology has now moved beyond amphibians. Researchers have successfully applied these same principles to human biology. These are anthrobots. They are self-assembling biobots made entirely from adult human tracheal cells. To test their potential, researchers placed a cluster of anthrobots onto a microscopic gap in a layer of damaged human nerve tissue. The anthrobots autonomously bridged the gap. By sitting across the wound, they triggered the underlying nerves to knit back together and heal. Since they are made from a patient's own DNA, these bots could eventually navigate the body without being attacked by the immune system, hunting cancer cells or clearing arterial plaque. This shifts the role of the doctor from a blunt interventionist to a collaborator, working with intelligent agents that can solve medical problems on their own. This raises a difficult question. What does it mean to release living, communicating entities as tools inside our own bodies? We are used to the risks of the microchip, an inert piece of hardware that only does what it is programmed to do. That biological hardware is different. It is inherently alive. It self-organizes, and it is capable of evolution. We have transitioned from building machines to choreographing life. We are left to wonder how long these organic creations will follow our lead before they begin choosing their own path.